I've had a really busy week this week. I've had a lot of interviews and appointments with bankers and accountants and fires here at the shop to put out, and it's just nothing really wrong or bad. It's just been busy, and I've not been able to produce a video for this Sunday morning. People that watch my channel know that I try to put out a video every Sunday at 7 o'clock, and not able to have one this week. So what I'm going to do is I'm going to pull a video from the past. It's, I don't know, 3 or 4 years old. It's an interview with Keith Deleplane, who was working at the University of Georgia at that time, smart man. It's about the polyandry and honeybees and how important that is. And it doesn't have a lot of views, which is interesting to me. I think it's because the title is not that appealing. Perhaps people don't think it's interesting. It was a great interview with Keith. He was very smart on the subject, and we went down a lot of rabbit holes, so to speak, that really some of them didn't even have anything to do with polyandry. There's a lot of good beekeeping information in this video. I recommend watching it. Older viewers of my channel would probably have already seen it, but it might be worth watching again. I don't know. I know I have a lot of newer subscribers that may not have watched my videos from several years ago, and I would recommend watching it. So tomorrow morning, Sunday morning, 7 o'clock, there will be a video with polyandry with Keith Deleplane. Keith, I really appreciate your time. I've known Keith a long time. I was thinking on the ride over here, I think I met you in the mid-90s somewhere, and I met you right here in the old lab. That's where I met you.
That's right.
So that's been a while.
There's a lot of beekeeping history right here on the UGA Bee Lab grounds.
Absolutely. It brings back a lot of memories from when I was a younger person. Maybe you were too.
I may have been
a little less gray. Anyway, so what I'm doing here today, I'm really keen on this concept of polyandry, and Keith is an expert at it. So I figured he'd be the right person to ask questions. And again, this is very casual. I'm not even sure. I should have a nice official list of questions to ask you, but you're so smart on this, I don't even think I have to go there. Polyandry. When I looked up the definition of polyandry online, it would show a picture of a woman surrounded by 4 or 5 husbands. So that's how when you bring up the word of polyandry, I think that's the concept that comes to most people's minds. But when we're talking about honeybees, it's something much different. I'm just going to let you talk. I don't need to say much here. Just tell us what you think we need to know.
Well, thanks for giving me the chance to talk about it, Bob, because I think polyandry is not only interesting from the perspective of honeybee evolution and why we have the insect that we know today. I think polyandry has a lot to teach beekeepers on how we can make our hives healthier and more productive. We are not really at a point yet where we can practically translate polyandry into practical beekeeping, but we're getting proof of concept that it's a thing that we must find a way to marshal and exploit and get it incorporated into our practical beekeeping. So it's a very fertile field, in my opinion, that needs a lot of work. But the promises are just are really sky high, and I hope to kind of convince you and your listeners today just how important polyandry is.
Well, you convinced me when I listened to you talk about it at the Clarksville Bee Club. That's why I'm here.
Okay.
I want to hear it all over again for the YouTube folks. So shoot, tell us about it.
Well, I think there's kind of a couple ways to go about it. You know, polyandry is important to the history of the insect over its evolutionary time. And there is a pattern that happens in biology where there are lower units that get sucked up into a bigger unit, and then those get sucked up into yet bigger units, and those get sucked up into yet bigger units. So it's kind of like a hierarchy. And at every stage we call that an evolutionary transition. And in this slide that I'll be showing you here, we have an example of that where you have like unrelated nucleotides just floating around in the primordial ocean, and how those are independent entities that reproduce on their own, and then they glom together into chromosomes. Well, then the chromosomes reproduce on their own, and then they get caught up into cells, and then the cells reproduce on their own, and then cells get attached to other cells, and voila, you have a multicellular organism. And that's the level where you and I are. We and all humans and organisms are multicellular organisms. And for each one of these steps to occur, there has to be a tacit agreement amongst all the lower units that, hey, we're going to suspend our selfish interests and reproduce together as a member of this new unit. And if they don't agree to that, that transition, by definition, can never happen. So there has to be this genetic agreement at each one of these steps. And so I'm interested in this process because it's general across all of life evolution, and it's especially highlighted in the honeybee. I think the cool thing, Bob, for your listeners and those of us listening who like honeybees and beekeeping, they are higher than we in this ladder that I'm describing, because we are organisms. Well, they are superorganisms. So in the case of honeybees and other superorganisms, you have individual organisms like worker bees who come together and they make that tacit agreement that they're going to suspend their selfish interests in the interest of the colony. So there you have it, one more step.
Well, it takes me into all kinds of ideas about politics and the world and the universe and so on. But
There is a lot of lessons there. And one of the things that's beautiful about evolution, a lot of times it is lampooned for being selfishness, unbridled selfishness. And people in our history in the last couple hundred years especially have chafed against it as a lesson in selfishness. But the flip side of that is cooperation. And cooperation is just as important as selfishness in the history of evolution. So we can never forget that half as well. So this is the big framework of everything to do with polyandry. And once you get to the superorganism, there's actually there's superorganisms that are simple and there's superorganisms that are complex. And in this slide that I'm going to show you right here, we talk about some of these differences. Simple ones have a single mother who's mated to one male, and they produce very small colonies. And examples of this are your bumblebees. Most of their females just mate with one male. And your wasps, many of them mate with just one male. And so you have they're a superorganism, but they're a very simple superorganism. There's not a lot of genetic stuff happening there. They're all super sisters with one another. Well, the flip side of that is the complex superorganisms. And with these, the mothers don't stop with just one male, they mate with multiple males. And this really upsets the formula because now you have colonies that are very genetically diverse, and it's no accident that it's in these complex superorganisms that we find the highest expressions of sophistication in the social insects. This is where you find the termites and the ants who rule planet Earth. And this is where the honeybees are. And it's because of the multiple mating habit of the females that they get different castes, different specialists, different all of the group decision-making behavior, the waggle dance, all of those sophisticated behaviors only happen in the complex superorganisms. The simple ones just don't have it.
So it's easy to get this concept in a beehive. You know, different colonies, for instance, will have maybe one's a good honey producer, one's really good at hygienic behavior, one's really good at defensiveness. And you can have all of those things going on in a colony at one time because you have that genetic diversity.
Exactly. That's a brilliant point, Bob. One colony doesn't have to be just one thing. Yeah. In fact, that's rather unlikely. It's very unlikely that a queen will have mated only with drones that are good honey producers.
Yeah.
But yet she's cranking out drones with her genes, and so you get colonies that have a little bit of everything, and that's the big advantage to the complex superorganisms.
Okay, so from a beekeeper point of view, I guess we're looking at the possibility, there's two questions here. We're looking at the possibility of a colony actually changing personality midstream over the period of a couple years and still have the same queen because she's mated with so many drones. So that begs the question, when a queen mates, is the sperm layering or is it mixing? Mm-hmm. Is she putting out all of these different characteristics at the same time? Or can she just put out one characteristic and then suddenly the colony changes personality in midstream and does something completely different?
That question is very persistent, and it has been around my entire career, and there's a reason for its persistence, and that's because in the scientific literature there's evidence for both. Most of the older literature, which is back in the 50s and 60s, had some evidence for layering happening, as you're talking about, so that a particular drone would be overexpressed for this period of time, for example, and then a different drone later on. But that model has been pretty much laid to rest and abandoned because the majority of later evidence says that it does mix. So the majority of our evidence is that the sperm mixes. And that makes sense from an ecologic point of view. If you think about it, if polyandry is beneficial, then you want it to be beneficial all the time.
Yeah, and of course, just looking in a colony, you can see the different bees.
Yes.
Some will be darker, some will be, we call it salt and pepper in a colony.
Right.
Both types of colors going on at one time.
Yeah, it's true. And there's 1,000 environmental reasons that can make a colony change its temperament.
Oh, sure,
too.
yeah.
So but the majority evidence is that the sperm is mixed.
Okay, so now, next question. If you've got mixed sperm and a queen, and you have a colony that you really like, and you want to use that queen to graft from, what are we looking at? I mean, you may not be getting the exact trait that you're hoping for when you pick that queen out as a breeder.
Absolutely, and Bob, you're kind of getting to the heart of the matter of what made me go into this line of research in the first place, and you're absolutely right. Let's keep things simple and say that an average queen mates with 10 males. Okay. Usually it's 10 to 20, but let's say it's 10. And let's say that you are interested in honey production. And you find this colony right here, by God, this honey doubled, tripled the yield over all the rest. I'm going to graft from this queen. Well, it's perfectly logical, isn't it? And so you go in there as a human wielding your grafting tool and you're filling your cups with the larvae, but it may be only one drone among those 10 who expresses the character that affects the entire colony that you as the beekeeper like. So on average, only 1 in every 10 eggs that you graft is going to be, carrying on the trait that you're interested in. So this is a built-in inefficiency in classical breeding with honeybees. And for most of my lifetime, honeybee breeding has been dominated by breeding models from other livestock, mammalian livestock models, which follow the paradigm that you just described. Hey, I like this cow. It's a great milk yielder. I'm going to propagate Bessie here because she makes more milk. But that's really iffy.
Doesn't work
with honeybees.
Heck, you can graft a bar, and every larva on there could have a different trait.
Absolutely. So it's got huge variation mixed into it. So, well, then take Bob the next step in that process. We have these virgins that we have grafted, which are already 1 in 10, let's say, that's got the brass ring. Well, now let's turn them loose and let them mate in the open, and it's kind of like a Wild West show. And so we should be amazed that we get anything at all accomplished in traditional honeybee breeding programs.
Okay, so I know you're an excellent artificial inseminator. So let's go down that rabbit hole.
Yes, I want to talk about that. I got a little image right here that you can show your viewers. And this is a typical Schley, S-C-H-L-E-Y, insemination device. It's what I learned from Dr. John Harbaugh when I was in graduate school at LSU, and it's been my most important tool my entire research career here at UGA. But this remains the only practical way that we can absolutely control mating number in queen bees.
So you still got to find that drone.
Yeah.
So does that mean that the genetics or that line of drones has to come from an artificially inseminated queen to get what you want?
If you really want to control this, yes. And here's often what happens. Classical queen breeders will make use of single drone inseminations an awful lot. So you raise a bunch of virgin queens and you inseminate each one of them with just one male, which is not natural.
No.
But that way when you get a colony that's expressing a character that you want, you got a really high probability that every egg you graft is in fact going to be carrying that trait. So it dramatically eliminates all that messiness in an open mated queen. So that's an important tool in bee breeding. So you can graft from these daughters from singly male inseminated mothers and improve that precision quite a bit. Of course, you still have the maternal side variation that's messing things up.
Yeah.
But it still allows for extremely well-controlled crosses.
So this is changing the subject a bit. I may be putting you on the spot and you may not have a good answer. When a colony inbreeds to a pretty good degree, you get all kinds of weird things going on. First thing that comes to mind is aggressive bees. What's going on there? Do you have a clue how you would get an aggressive colony from inbreeding? I've seen it a number of times. Inbreeding, in my view, can cause aggressiveness.
If you have a lot of inbreeding going on, and if you have aggressive alleles in your population, they are equally likely to be among the alleles that are expressed dominantly. Dominant, on either side of the chromosome in your workers. And so if you have aggressive alleles in your population, and if that population is inbred, then that could be one of the characters that is overexpressed. They're just nasty. Equally, you could have really bad thermoregulators. The opposite could work. You could be getting overexpression of negative traits or recessive traits, mutations that are non-productive. That's what inbreeding does. It exaggerates those alleles that are the same on either side of the mother-father divide.
You remember the star line, midnight line?
Yes.
That was very interesting. My understanding was they would inbreed them to the point of near collapse.
Yes.
And then take two separate lines and breed them together and come up with an extreme hybrid vigor.
Yes.
I never could quite wrap my head around that, why you could take two really, really bad lines because they were so severely inbred, then suddenly come up with a queen that was really superior. Mm-hmm. Of course, you'd lose that the first time that colony swarmed or superseded. That hybrid vigor would disappear. Are you able to explain this a little bit?
Yeah, well, what you get, and this principle applies to hybrid crop production as well. Hybrid corn, for example, employs this same method. They will inbreed different parental lines, and these parental lines are just pathetic. I mean, they're weak, spindly little ears of corn.
Just like the bees.
Pathetic, yeah. Just like the bees. And what inbreeding means is that the individual has the same form of the gene from its mother and from its father both. And nature in general does not like that. If you have an individual up and down its chromosomes that has the same form of this gene, same, up and down the chromosome, it's almost a common law of biology in animals and plants that those individuals are very poorly productive. They're not vigorous. They're very immune. They're very prone to other kinds of maladies or disruptions or threats. They don't have the genetic diversity. So if you have two parental lines in which every form of the gene is the same, with the male line, and over here the female, it's the same but different, a different form, but same. And then their gametes form, cut in half. You got the egg over here, the sperm over here, and you bring them together, pow! Now what do you have? You got different. You have the exact opposite extreme now. Every gene has the opposing form of that gene from the mother and from the parent. And this is what a hybrid is. Maximum diversity at every gene locus.
Okay.
And nature loves this.
Okay. Now let's go down another rabbit hole. Haploid, diploid.
Yeah.
How many creatures on planet Earth do that?
There's quite a few.
Is
There are surprisingly quite a few. They are overrepresented in the arthropods, which include the insects. In the order Hymenoptera, which includes the ants and the wasps and the bees, it's universal.
Really?
All males in the ants and the wasps and the bees carry just one set of chromosomes. It also occurs in, coincidentally, the mites that we're familiar with.
Oh, really?
Tracheomites have a
Well,
haplodiploid system.
Yeah, that's an interesting thing. Constantly inbreeding.
Yeah.
That's another thing that I don't quite get a handle on.
Well, there are exceptions. And that's the thing about biology, it loves exceptions as well. And there are times in species for which inbreeding is not a problem. And wouldn't you know that our chief nemesis, the Varroa mite, is one of them. It can handle enormous levels of inbreeding with no apparent loss of vigor whatsoever.
How many, I know we're here talking, forgive me, my mind just comes up with all these questions as you're talking. How many, I don't know if species is the right word, how many different Varroa mites are there?
I don't know offhand. I'm thinking there's in the neighborhood of 5 or 6. There's roughly 20 species of Asiatic mites of the genus Apis. So there's not only other Asiatic mites, there's other species of honeybees, Apis, concentrated in Southeast Asia. And they all, every Apis in Southeast Asia has its own co-evolved parasitic mite. Varroa is the Varroa destructor that we're most familiar with as honeybee keepers, is the natural parasite of the honeybee Apis cerana from Southeast Asia. And through human trafficking, it's been spread throughout the whole world now. This is another conversation in its own right, Bob.
Yeah, I know. Getting off subject. But...
well, it's not irrelevant. Honeybee, the western honeybee that you and I keep, alone of all the genus Apis, it is the only representative of its genus that has no Asiatic mite, which is strong argument that the lineage of Apis mellifera never evolved from Southeast Asia, and that it is its own independent lineage, probably evolving in Europe, because it has no history whatsoever of an Asiatic mite. And this is an important point. It's a little bit down a rabbit hole for our purposes right now, but actually I can kind of loop back and talk about this.
Help yourself. Like I said earlier, we can't see anything wrong here. Just have at it.
Yeah, but it's important to realize, as beekeepers do, that genetic resistance in honeybees against Varroa mite is the holy grail.
Absolutely.
That's what we're all wanting and working for. I think it's important for us to temper those expectations with the knowledge that mellifera has never, ever, ever had an Asiatic mite in its lineage. So when we come around in the 20th and 21st centuries and we say, I'm going to breed a Varroa resistant honeybee, the question is how? With what tools? With what genes? The insect doesn't have them. Where are they going to come from? Are you, Mr. Breeder, going to invent new genes? No, that's not what breeding does. Breeding resorts genes and concentrates them in individuals and in lines, but it doesn't create new genes. So whenever we do find lines of honeybee that express resistance to Varroa mite, and they do exist, we all know about this. Yeah. When we do find that, it's important that we recognize that the honeybee is co-opting a different tool to a brand new job. In the case of Varroa sensitive hygiene, for example, the hygienic behavior, which is natural to Apis mellifera, is an evolved response to brood diseases.
Yeah.
And it has just been sort of tweaked and shaped and formed and edited to be effective against Varroa mites in brood. And it works to some degree.
But you lose it quickly. Like
you do.
If you I'm keeping it simple, but if you were to buy a Varroa sensitive hygienic queen from somebody, bring it into your outfit, the first time that colony swarms or supersedes, what happens? You lose a lot of it, is my understanding.
That is very true, and Bob, you are just a fountain of talking points, because there's a reason for that. There's a reason.
Explain the reason.
There are, and it's intuitive when you think about it a little bit. It's the principle of too much of a good thing. And there is a principle in animal breeding that if you have alleles, which is simply a form of a gene that's out there in your population, that are outrageously beneficial for a particular thing, that nature has evolved for those super beneficial traits to be rare and recessive. And if your viewers can remember your high school biology or college biology, there are forms of genes that are dominant and recessive. And so these highly specialized alleles have been evolved to be rare and recessive, which means it's harder to express them. The mother has to have it, the father has to have it, in order for that double recessive to be in the child and for that benefit to be expressed. Why would they be rare and recessive? It's beneficial. Why in the world should nature evolve for these hyper-potent beneficials to be rare and recessive? Well, you can have too much of a good thing. You can get that trait locked into a breeding population so that it is extremely good at resisting that one potent thing to the cost of everything else. And I can give you a good example. Hygienic behavior can be overexpressed.
Yeah, I know that, yeah.
Let's say, for example, well, hygienic behavior, it's beneficial, right? You know, you want your bees to have it. But if you have it locked in your population so that every drone and the queen that he's mated to, they're all expressing hygienic behavior, all of the workers are dominant for recessive, for hygienic behavior, you will have too much hygienic behavior. You will have nurse bees that are going around aborting and jettisoning perfectly healthy brood, and this happens.
Well, occasionally you hear about these bees being non-productive. Mm-hmm. Is that the simple reason in a nutshell?
It's certainly part of it. Overexpressing a good trait. Mm-hmm. I think of these beneficial traits as sort of like Tabasco sauce. You want a little bit of them scattered throughout your worker population, but you certainly don't want to drink the whole bottle.
Okay.
And this kind of ropes us back into our original topic about polyandry.
Okay.
And it is very likely that one of the evolutionary drivers that pushes these females to be promiscuous, to molt with many males, is to cast a wide net, not only that she picks up that drone over there that's carrying that beneficial gene, but also that she mates with the other 99% who don't have it. It's the flip side of that, because it's equally important that she not overexpress that really beneficial trait in her colony, or else it will become ecologically non-competitive. It'll be really good at resisting Varroa mites, and maybe really, really bad at thermoregulation, or pathetic at foraging for nectar or something. It'll be too specialized. And because it's so potent in near time, it gets rapidly fixed, and that's a danger. Again, nature, on average, prefers diversity over specialization.
Polyandry, that's an interesting word. I would challenge everybody to look it up online and look at all the different definitions of it. It's a crazy word.
I want to tell you, I think it's we would be remiss, Bob, to not talk about some of the work that we have done that I think has real practical application to beekeeping.
I was going to go there with you. I didn't know if I'm allowed to ask you questions about the new vaccination. Is that where you're going?
No, I'm still not done with polyandry, but then we'll get to the vaccination.
Okay,
because I know your listeners are curious about this. And want to hear about
it's a biggie right now. Go ahead.
I want to talk a little bit more about the use of polyandry to solve the genetic problems that we're dealing with in beekeeping today. And I have done quite a bit of work the last 10 years on trying to translate polyandry into practical stuff for beekeepers. And what we did, I want to show you a slide right here that kind of, I think will be helpful to your audience. We did an experiment when I was living in England on sabbatical where we inseminated queen bees with 3 different numbers of drones. We inseminated some of them with 15 males, some of them with 30 males, and some of them with 60 males. And these males were totally unselected. They were just a random drones from any particular colony. And then we reared these queens, we inseminated them, established them in colonies, and then we measured them for brood production and their mite loads. And what you're seeing here is a significant increase in brood production in those queens that were inseminated with 30 or 60 drones, above and beyond those that were inseminated with 15. So these are significant differences. So over here, we now have the same numbers for mite drop, and the bars are going in the exact opposite direction, which is good, because there was significantly more mites in the queen's colonies that were inseminated with only 15 males, and it was significantly lower mites in those queens inseminated with 30 or 60.
That's interesting.
Now, these are 2 of perhaps the most important economic traits for beekeeping. Big populations, low Varroa mites. I mean, what more could you want? This is great stuff. And this is a perfect example of what you can get with polyandry in the complete absence of breeding. I think of polyandry as almost anti-selection. We did not breed for drones that produced high brood. We did not breed for drones that produced low mite levels, but simply by filling our queen's spermatheca with sperm from a lot of males, we got both of these economic benefits.
Okay, so let's put this into a practical application. I read an article a long time ago, probably 30 or 40 years ago. It was a question answer in one of the bee journals, and the question was, how many support colonies should you have producing drones for a certain amount of virgin queens? And this man answered 6 support colonies for every 100 queen mating nukes, and I thought, that doesn't sound like enough. I've always been a proponent of the more drones the better. You just get better queens. Just looking at the colonies, it's obvious that what you're getting is something better. So if you've got several hundred queen mating nukes out in the pasture, I like to say we try to surround them with 3 very populated bee yards. We're getting a lot of drones in the air, millions maybe. I don't know how to calculate that. We always have better queens. We always have less supersedure. So that's kind of the practical application of this idea, isn't it?
It is huge. It is huge. There is one more that I will get back to here in a minute, but I want to pause on this one since you brought it up. The number of matings in a drone congregation area, generally range from 10 to 20, but it's also, there's evidence, my colleague and friend Dave Tarpey at NC State has done some work on this, and he has shown that a lot of times the numbers are just random, that it is so chaotic when you are a virgin queen flying through a comet of young randy drones. The numbers of copulations can happen breathtakingly fast. And I know this in part from my work at LSU. We would go out and study drone congregation areas, and you could hear them exploding in the sky.
Oh, really?
Like popcorn.
I've heard that. I've never heard it.
Yeah, and they'd come raining down on top of you. But
I've heard of that.
But the mating can happen just pow, 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 really fast. And so a lot of times a queen can just accidentally end up getting inseminated of up to 70 drones. And Dave's group has in fact shown, Dave Tarpey's group possesses the world documented record for the number of natural matings, and it's over 70.
Wow.
They have found queens mated with over 70 drones, and it's very likely that it happens just out of randomness.
So they found sperm from 70
Yes.
drones in a queen.
In a single queen.
Wow.
So this supports what you're saying. We want huge drone congregation areas to allow those happy accidents to occur with greater frequency.
So when I'm talking about queens to a group of beekeepers, I kind of beat up on some of the big queen producers in South Georgia and other areas, how they're trying to get these queens out early. And the bees really do have a sense if the queen is not mated. She might be well mated with a few drones and have a good brood pattern, and she's accepted and she does a great job, but we find this early supersedure. I'm thinking the bees have a sense that she's not mated with a lot of drones and they want to replace her. Am I realistic
That's true,
with
but
that?
the queen herself also monitors this. There's no evidence that queens can count. And experiments have tried to test that. Maybe she's keeping a count. She has an image, an idea in her mind of how many matings she needs. The real driver is more likely the volume of sperm that she feels in her median oviduct, because she does have a nerve supply to the median oviduct, and she can tell when she's distended with a lot of semen.
Well, how does that relate to the colony wanting to supersede her early?
Well, sometimes she may adjust her expectation downward if there's not a lot of males in the neighborhood. And they have shown this with research in Poland, that a queen where there's lots of males, that queen's threshold for how tight she wants to feel in her median oviduct is higher than if she's in a neighborhood with fewer drones. In other words, she lowers her expectations. And she says, well, I'm not encountering very many males, so I guess I'm just going to quit. I guess this is as good as I can do. And she goes back to her nest and she's done. So this reinforces the importance of huge drone congregation area populations. You want her to just be pow, 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 encountering lots of males.
Can I drag you down another subject? And you may not have an answer for this, but there seems to be some research showing that when a queen lays less eggs, in other words, a queen in a small colony, Brother Adam was all about this, keeping your breeder queens confined to a smaller unit. He felt like the eggs were bigger and better and would produce better larvae if the queen was restricted in her egg laying. Do you buy into that, or do you have any information on that?
I can't say that I know about that, and I'm groping to understand a mechanism. What would make them the eggs?
They're able to be. I'm guessing maybe a little more nutrition in the egg because she's not producing so many.
Okay. Right. Well, I mean, that sounds plausible, but I don't know, Bob.
Okay.
That sounds
I stumped
like a good
Keith.
guess.
I didn't know I could do that.
Trust me, that's easy to do. Well, before we leave this subject, before we talk about vaccines, I do want to make your listeners aware of one more way that we can approximate polyandry, and that's through brood mixing. And I have 2 slides here that I'll share with you and your viewers. Let's say you have 3 colonies here that are coming out of winter, and there's the brood combs. Well, you can redistribute those combs like this, so that you have one of each brood comb now scattered amongst these 3 colonies. And the premise here is, if a queen, once again, just keep things simple, if she averages 10 matings, we could say that each one of these queens is mated to 10 males. So then if you swap the brood around, you have a temporary flush of workers that now represent 30 males on average in each of these 3 resulting splits. So this is a method that we have been studying to try to imitate or simulate the effects of polyandry, and we've got some good results. I think you have interviewed Lewis, my new colleague.
Yeah, Lewis is great.
Yeah, well, we did a study where we mixed brood in colonies in this manner to make an artificial polyandrous cohort of workers, and we also fed them traces of pollen with neonic, and some of them with control pollen without any neonic. And we were interested in how the effects of mixing could mitigate the effects of neonics. And here we show these. This is the scatter of the data, and this is brood survival, how well the brood survive. And these 2 bars here are of interest. These are the colonies that received mixing without neonic and mixing with neonic. And in both cases, the effective mixing was strong enough to elevate brood survival, even in the presence of neonic. So this suggests that even in a simulated polyandrous condition, we can effect some of its benefits.
Can you explain that?
It's a temporary expression of polyandry.
But I get that, but can you explain these results because of polyandry?
My guess would be we now have 30 different genetic lines in each of these colonies instead of 10, which means
gives you the possibility of another another line or two that's able to
Yes.
survive or overcome.
Yes. It's
Okay.
a probability game. We are increasing the probability that there are patrolines there that are able to detoxify foreign substances, or able to remove and recognize foreign substances, or remove themselves from nurse duty altogether because they have been subjected to an insecticide. You get 1,000 different genetic possibilities, but the point is we have given these colonies more genetic variation than these.
Okay, this subject, a little different. Explain to me, I believe that when I mix brood in a split, I have better queen acceptance. Can you explain that to me?
It's possible that you are increasing the principle against nepotism. When you have a highly related cohort of workers, there's always the danger that they will be more particular in the type of queen that they accept or that they feed. But if you have a genetically diverse workforce, that effect of nepotism just gets diluted away.
Makes sense. Perfect sense, yeah.
That would be my guess.
Yeah. There's a lot going on here that we don't understand.
Well, here's another thing that excites me about polyandry, and Bob, why I think we need to mainstream this into bee breeding. It is conceptually so simple.
Yeah.
So simple. Think about it. Classical breeding, you have to select a colony, you have to propagate the virgins, you have to try to control the drones they mate with and hope for the best at every step. And you might get 10% of your product that's actually expressing the product at the end of the day that you were hoping for in the first place.
Yeah.
It's outrageously inefficient. Polyandry is the conceptual opposite of that. It is anti-breeding. We are selecting instead for queens that carry the maximal different versions of each gene that we can possibly cram into their spermatheca. So it's easy. It's easy. You can inseminate them, you can mix brood, you can promote big drone congregation areas and mimic the same results that are achieved through heroic efforts in classic breeding. So I consider it a win-win, and I encourage breeders and beekeeper groups to get more familiar and comfortable with instrumental insemination. You know, it works. I'm interested in the possibility of breeding for promiscuous queens.
Absolutely. In fact, I meant to ask that question when you were talking, and it slipped my mind.
And it'd be conceptually easy to do. You could, just simply inseminate and rear a bunch of virgins, and then, genotype their worker offspring. It's non-lethal. You're genotyping her workers, not her.
Yeah.
And continue to select from those lines that mate with the more males. And I think it's kind of fun too. Think of the marketing possibilities. What name would you call that queen line?
Yeah.